What's happening, guys? Mr. Rouge again, and this time we're talking about enzymes, which are known as biological catalysts. So let's take a look at what these are and why they're important. Well, I've showed you guys this picture before, and this is of your metabolism. This is all the chemical reactions that are going on in all of your cells. Now, what's controlling and what's allowing this metabolism to happen are enzymes. And I've mentioned in the past that you need vitamins and minerals to work in your metabolism. Well, those vitamins and minerals are going to work with enzymes, okay? So the enzymes are what's doing all the work. The vitamins and minerals are helping them do their work, okay? But what's controlling all these reactions are enzymes. So let's take a look at what enzymes are. Enzymes are proteins that speed chemical reactions. We use this word catalyst, and that word to catalyze means to speed a reaction, okay? So if I say enzymes are a catalyst, that means that they're speeding up the chemical reaction. We need to know this word, substrate. The substrate is whatever the enzyme is working on, okay? And you guys will see an example of that in just a moment. The active site is where the substrate binds to the enzyme, okay? And I think of this as a key and lock model, okay? So what's going to happen is when the substrate fits to the enzyme, it's going to fit perfectly like a key fits into a lock, okay? And we call that the key and lock model. After enzymes go through their chemical reaction, they remain unchanged. So one enzyme will continue to work after the chemical reaction, and it's going to continue to find more substrates. Most enzymes end in the suffix ase, similarly to how sugars ended in the suffix ose. Please don't get those mixed up. Ase means enzyme, ose means sugar, okay? Ose is sugar. Enzymes need a specific temperature and a pH to work. If the temperature gets too hot, or if the pH drops too low or rises too high, the enzyme becomes denatured. What that means is it's lost its shape from the pH or temperature. If it doesn't have the right shape, it doesn't work. It won't be able to function, just like we go back to the key and lock model. If I change the shape of a key, it will no longer work on the lock, okay? Now there's two types of reactions. There's an anabolic reaction. That's where the enzyme is going to build something. And yes, it's going to go through dehydration synthesis. That's one anabolic reaction. So anabolic just means the enzyme is going to build, and it can be achieved by dehydration synthesis. A catabolic reaction is where the enzyme is going to break it apart. So think of hydrolysis, okay? So this reaction right here, you can see that there's two pieces to the substrate. They fit to the active site, and when they're done, the product is one thing bonded together, okay? If we look at a catabolic reaction, this one right here, it's taking in the substrate and then splitting it apart. So go ahead and take note of all of this. Okay, there's not a pencil here, but there we go. That's our pencil. Go ahead and take note. And give these a quick sketch down at the bottom. So we have our enzyme. This area right here where the substrate fits into, that is the active site. Again, the substrate is whatever the enzyme is acting on. When they fit together, we call that the enzyme substrate complex. When they're done, the remaining parts of the reaction, what's finished, are called the products, okay? Please make sure to give these right here a little sketch, okay? And take note of everything on this slide. Okay, so let's take a look at how enzymes work in a normal temperature and a normal pH. So we have our pH scale down here at the bottom. We have our temperature scale down on the side, and there will be an optimal range where the enzymes work best. If we're in that range, we're okay. So we could see the enzyme is going to the substrates, they're binding to the active site, and it's splitting them apart. It's going through a catabolic reaction. Okay, And we can see that the enzyme is continuing to work on more than one substrate. So after each reaction, the enzyme isn't changed. But now what happens if we raise the temperature, we get too hot, and we drop the pH, it becomes very, very acidic. Look what happens to the enzyme. The shape has been altered, okay? And think back to the key and lock model. The key has been changed, so now it will no longer work on the lock. The enzyme will no longer work on the substrate, okay? It cannot fit to it and does not work. So temperature and pH are very, very important. And I get this question sometimes. What if it gets too cold? Here's what cold will do. If I drop the temperature, it's not going to change the shape of the enzyme. We're just going to have a much, much slower chemical reaction, okay? So the enzyme is still working. The shape is still the same. It's working on multiple substrates, but it's just a very slower reaction, okay? Again, if I were to increase the temperature or increase the pH or decrease the pH beyond what the enzyme needs, then that will denature or change the shape of the enzyme, okay? Let's just review. And let's do a quick recap. So, enzymes, they are proteins. They are not carbohydrates. They are not fats. They are not nucleic acids. They are proteins. They catalyze reactions, which means they speed them up, okay? Most enzymes end in the suffix ase, okay? So like we'll hear names like catalase or polymerase or helicase. We all know those are enzymes because of their suffix ase. After each reaction, they remain unchanged, and they continue to work on more substrates. The shape allows them to do work. If it's denatured, that means the enzyme won't work. It has lost its shape. Just like if I change the shape of a key, it will no longer work on the lock. pH and heat, that is what will denature the enzymes. Cold temperature will slow the reaction down. It'll still occur, but just much slower. And enzymes can build molecules, which is anabolic, or it can break apart molecules, which is catabolic. And if we look at the parts of an enzyme and substrate, the active site is where the substrate binds to. So we have our enzyme here. We have our substrate. The substrate is going to bind to the active site. Once they're together, that is called the enzyme-substrate complex. And when we're done with the chemical reaction, we're left with the products, okay? That is it for today, folks, and I'll catch you all next time. Adios.